After the genome assembly, we get the chromosomes or pseudochromosomes or contigs, and our next job is how can we find different number of genes. So in order to do that, first we see how we define genes. But there are some issues that defining genes is problematic. It's not an easy task because small genes can be difficult to detect. One gene can code for many proteins, and then there are some issues like some genes can code only for RNA, and sometimes two genes they can overlap, and they can produce a chimeric product. So these kind of issues, they make this situation really complicated. How we define genes, this is not the exact, but I would say the most appropriate definition which I get it. So gene is a piece of DNA, a discrete unit of genetic information, which encodes RNA or protein polypeptide molecule performing some function. So that function is performed in the cell alone or together with other RNA or protein. So in this definition, we can see that the gene encodes for a protein or RNA, and there is also some functional implementation of that product. If we look into the historical viewpoints, it looks like a tangled knot. So somewhere in the history, they defined that one gene that encodes for one function. And later on, that definition changed that because one function may be a combination of different enzymes, and they have seen those genes controlling different enzymes. So the next hypothesis came was one gene, one enzyme. Then later on, the enzymes, when they were studied, it was found that they are made up of different polypeptides. It was hypothesized that one gene may be associated with one polypeptide. And then later on, that came to one gene play a role in formation of a number of polypeptides. And now we see that even one polypeptide is associated with a huge number of genes. So in this way, number of genes can give you one polypeptide. So defining gene has always been a cumbersome task in the history. If we look into the splicing patterns, the genes, when they make their products, they have in between them, they have their introns. So different genes, then they can combine with one another. And if we look into this alternative splice patterns, their exons, they are spliced alternatively. We have seen that in some previous sections, so I'm not reinforcing that concept. So here in this slide, there are different ways in which we can get these alternative splice variants. Sometimes we have mutually exclusive exons, so some exons they are present, others are not there. Sometimes we have an intron retention where introns are retained, and even the alternative 5' or 3' sites, they are alternatively used. And somewhere we use 5' alternative splice site, and sometimes we have the 3' coming from the other exon. So there are different scenarios in which we can see there are different forms of alternative splicing. So all these things, they make it's really cumbersome task how to exactly define gene and what patterns we should look into in order to identify the genes from a sequence. Here is a eukaryotic situation. In this slide, we see that there is some gene 1, so that have different exons which code for proteins actually, and they are separated by introns. So here is its promoter. We have intron 1, and then we have exon 2 with, and then 2 and 3 are separated by intron 2, and then in between they have some intergenic region, and again there is a promoter for another gene. So this is how these genes are arranged on the chromosomes in a eukaryotic cells. So both of them, they can use their own promoters. Sometimes a single promoter can go and transcribe two genes together. So in this way we can get a combined product of both the genes, a chimeric product. This process of formation of RNA is transcription. So when we make that RNA, say for example in this case we make this long RNA from this promoter number 1, and this is kind of the situation which is there in the prokaryotes. So genes are transcribed by a single promoter. So we get that pre-messenger RNA. Here obviously we are looking into a eukaryote because we have exons and intron situation. There is some alternative splicing, and we get exon 1 in the final product and exon 2 of the gene 1, and then in the mature messenger RNA we get exon 2 and 3 from the second gene. So in this way this RNA is kind of chimeric RNA, so that is a combination of both those genes, and that might lead to a chimeric protein. So this is how we can see that we can combine two genes and that situation. So if you are actually trying to get the genes from their products, so those products may be coming from different genes, so it's a cumbersome task. It's to identify the genes. So about 4 to 5% of tandem gene pairs, the gene pairs which are located together, they are estimated to be transcribed into a single messenger RNA. So that is the situation where the chimeric products are there, and these kind of events make it hard to do gene prediction. So gene is a DNA sequence of DNA that is transcribed to produce one or more functional products, and we see in this section that defining gene is a hard task, and it's a difficult task due to issues with complicated gene structure and function.